So welcome back to Chapter 3, Part 2. In this part, we're going to focus on carbohydrates. Carbohydrates are anything that really serves as fuel and building material for your cells. And really, carbohydrates include things called sugars and anything that's a polymer of those simple sugars. The simplest carbohydrate is called a monosaccharide. Mono, again, meaning one, and saccharide meaning sugar. So we call those simple sugars. Carbohydrate macromolecules are polysaccharides. Poly means many, and polysaccharides are polymers made up of many simple sugar building blocks. Monosaccharides are usually some version of a formula that goes by CH2O. So those are going to be our simple monosaccharides. For example, glucose is C6H12O6, which is a multiple, right? If it's like saying 6 C's, 6 times 2 is 12, so 12 H's and 6 O's. There it is, glucose. Glucose is the most common monosaccharide, but there are others out there that we'll be talking about very briefly. And monosaccharides are classified by the number of carbons in the carbon skeleton. So, and wherever the carbonyl group is placed, remember, the carbonyl group is the C double bond O. So let's take a look at some monosaccharides. We've already mentioned glucose, right? Here's glucose, and there's that carbonyl group. So this is glucose, 6 carbons, and if you count them up, there's going to be 12 hydrogens and 6 oxygens. This is fructose. Fructose also has C6H12O6 as its molecular formula, but the location of that carbon double bonded to that oxygen is different. And so that gives that functional group has moved, and that gives it a different function. This fructose is very different from glucose, even though it looks so similar to us. Let's take a look up here at a simpler monosaccharide here called glyceraldehyde. It's 3 carbons, and there's our functional group with that C double bonded to that O coming off the end. This is a pentose sugar. This is probably one of the most important pentose sugars, or 5 carbon sugars. 1, 2, 3, 4, 5. This is ribose, and ribose is a component of RNA. We'll be talking about ribose and deoxyribose later. Deoxyribose has one of these oxygens missing, so we'll talk about that later. But for now, notice the formula is C5H10O5. Sugars are often drawn as lines or linear skeletons. But in water, in aqueous solutions, we say, in water, sugars tend to form ring structures. And a lot of the time, when we're showing you sugars, we are showing you them in the ring structure because for the most part, these are existing in living things. And of course, living things are filled with water. So here's what happens when we form the ring structure. The 6th carbon or the 5th carbon will join up to the 1st carbon. And the one that we're going to show you, it's going to be the 5th carbon that's going to join up with the 1st carbon into a ring. And so kind of pay attention to how this happens. We have this folding over. And so the 1st carbon comes into contact with the 5th carbon. Oh, and let me just back up a second and say that we number our carbons based on whichever one is closest to that acid group. So, I'm sorry, the C double bond O group. So here we go. That's the 1st carbon. That's the 1st carbon. And then counting around in the circle this way, or counting down the line this way. So eventually we're going to form a bond between the carbon and that oxygen. And notice when this bond forms, the hydrogen moves down to here. it still has the same number of carbons and hydrogens and oxygens. They're just conformed slightly differently. Also notice that when you look at things in a ring structure, we number the carbons 1, 2, 3, 4, 5, and 6, but we don't show the carbons. So anytime you have an apex here where two lines meet and there's nothing written there, that's a carbon. So there's a carbon there, and there, and there, and there, and there. You can see it in this structure. We call this the abbreviated ring structure, and it really becomes important when we've got a very busy molecule that we're looking at. Now we form disaccharides, di means two, when you've got a dehydration reaction and it joins two monosaccharides. So here we are, dehydration reaction, and this is glucose and this is fructose, dehydration reaction right there, and our water is released, and we have a link, a covalent bond link between two monosaccharides. And now we call this molecule sucrose. Sucrose, that's table sugar. The link that forms between the two sugars is called a glycosidic linkage, and that's a term that we'll be talking about quite a bit in this semester. So this is called a 1,2 glycosidic link because it's between the first carbon of glucose and the second carbon of fructose. 1,2 glycosidic link. Polysaccharides are polymers of sugar, and they are really important in storage and structure. Poly just means many, and we don't need to know exactly how many each of the polymers has, although if you have extra monosaccharides in a polysaccharide, then it's going to have a different name than if you have fewer, right? The structure and function of polysaccharides are determined by, number one, what the monomers are in there. So is it glucose, is it fructose, is it galactose, and the position of those glycosidic links. So some of the polysaccharides can be important in energy storage, and those are starch and glycogen, which we're going to talk about in a minute. Or they can have structural function, like cellulose and chitin. Yes, oops, sorry about that. This word is chitin. Whenever you see a CH at the beginning of a word in biology, it sounds like a K. So we're going to go through each of those and talk about them a little bit, talk about their chemical structures and their roles in cells. The first one is starch. And remember, I just told you that starch is a storage polysaccharide, which means that it's important in energy release. And starch is found in plants. It's made up entirely of glucose, and it's made up of the same type of glycosidic link. So if you remember the counting, this is carbon 1, 2, 3, 4, 5, 6. So this is a 1, this is 1, right? And this is 4. So this is a 1,4 glycosidic link. Sometimes that gets the symbol alpha. Plants store their extra starch in granules inside of the cells, and when they need that energy, they'll break off glucose molecules. Animals that eat plants can have enzymes that break down those glucose bonds, those glycosidic links, and so it makes that glucose available as a nutrient in the animal. Glycogen is an important polysaccharide in animals, and one of the big things that we see in glycogen is that the way everything is linked is different from the way that starch is linked. We've got a little bit more variety, we've got more branching. We find lots of glycogen in the liver and muscle cells because the liver and muscle cells are probably doing the most work in your body. We'll get into what the liver does later, but you already know what muscles do. So yeah, lots and lots of energy needed. That means we need lots of glucose, and we're storing that in the form of glycogen. Just a quick look here between some of the differences between the three main polysaccharides in plants and animals, and the starch, glycogen, and cellulose. I've already talked a little bit about starch and glycogen. Remember, starch is a storage molecule for energy in plants, and we talked about the glucose monomers. Glycogen is the granule that's stored in muscles and in liver. Lots of branching going on here in glycogen. And then there's cellulose, which we haven't really talked about very much. We'll talk about it a little bit more in the next slide. But one of the very cool things about cellulose is because of the way that it links those glycosidic links, bonds, these molecules are very flat and they're unbranched. Remember that those dots are hydrogen bonds. So what we have in glucose is a lot of hydrogen bonding between molecules of cellulose, and that makes them very, very, very strong and also kind of parallel or flat. So here you see the very parallel and flat links in the cellulose. Let's take a closer look at cellulose. Cellulose is a major component of plant cell walls. Those are the things that enclose plant cells. They're on the outside of plant cells, and they really give a lot of structure. The glycosidic links do differ in those, as I showed you before, and a big part of that is the difference in the ring structures for glucose. So you can have alpha glucose or beta glucose, and that tells you which version. Now, quick remembering here, we've got the oxygen in a different place. Do you remember what kind of isomer those are? If you don't, rewind and look back and see if you can figure that out. So just because we have the different ring structure, the alpha and the beta, we have different ways that they can string together. For starch, we really have a lot of alpha glucose molecules hooking together in that 1,4 link that I was talking to you about before. In cellulose, we also have a 1,4 link, but those are linkages of beta glucose monomers, and those beta glucose monomers, we have them upside down with respect to its neighbors, so we've got them on different sides, and that's what gives that cellulose a really flat nature. Our bodies cannot digest cellulose. This is a really important thing because cellulose is also known as fiber, and since our bodies can't really break down fiber, this helps with the digestion process. It keeps our whole guts, flowing freely, and it helps loosen things up because it's not digested by our bodies. Some bacteria that we live inside of our guts can break down cellulose, but our bodies can't do that themselves. So in starch, the glucose monomers have the alpha conformation, and starch molecules can be helical, and all that helical means is that they are in kind of twisty, turny, kind of like a slinky shape. If you remember playing with slinkies when you were a kid. Cellulose have the beta conformation, and those are straight and unbranched. Again, just focusing here in on those hydrogen bonds that hold across different cellulose molecules. They're really strong. They really help keep everything in place, and so that gives that strong building material for plant cell walls. Enzymes can break down alpha linkages, but they cannot break down those beta linkages. And I talked about this in previous slides, but I just wanted to make sure you had the words there for you. So cellulose does pass through our digestive tracts as insoluble fiber, but as I mentioned before, some bacteria can use their own enzymes to digest cellulose. So what's really interesting is that even if we think about things like cows and termites, they also cannot break down the cellulose molecules. but that's all they're eating is things that have a lot of cellulose. So herbivores like cows and termites rely on this symbiotic relationship with microbes. Symbiosis just means that there's a close biological relationship. It means that they live very closely. In this case, the microbes are living inside the guts of the herbivores. Now, fungi, those are mushrooms and things along those lines, they can digest cellulose, and that's really important in helping recycle the Earth's elements and making ecosystems kind of balanced, and so that's an important role of fungi. Now, we were just talking about fungi, and I don't know if you remember, but there were actually 4 polysaccharides, and one of the 2 structural polysaccharides besides cellulose is chitin. So chitin is found in 2 main places in living things. It's found in the exoskeleton of arthropods like insects, spiders, those kinds of things. It's also found in the structural support in the cell walls of fungi. So here we have chitin. Now chitin is that ring structure, right? It's definitely going to be a saccharide. This is a monosaccharide right here. This whole thing is a polysaccharide. But notice that the functional group that come off of those monosaccharides has some nitrogen. It's a little bit more complex there. And so that's the important ingredient that makes these monomers different. All right, that's the end of chapter 3, part 2. We're going to start up with chapter 3, part 3, learning about lipids in the next video.